Good morning, everyone. For this new episode on HLA this time, we are from TransConnect. This is February 2023, and I am Dr. Mohit Chaudhary, a transplant immunologist in New Delhi. The topic for today is single antigen bead assay and virtual HLA crossmatch. This is a very important topic, especially for people who are working in transplant immunology lab. But before we start this episode, you should be aware that we have a episode already on HLA overview, the basics of HLA. So please go through that first, and if you have gone through it, it will be easier for you to understand what single antigen bead assay is and how we perform this virtual HLA crossmatch. So coming to the basics, MHC and HLA typing, we all know MHC and HLA are interchangeably used. MHC is major histocompatibility complex and HLA stands for human leukocyte antigen. So MHC or HLA is located on the 6th chromosome, small arm, 6p21.3. It is divided into three regions, the class 1, the class 2 and the class 3. Class 1 and class 2 are more important to us, especially in transplant immunology, because they are very polymorphic and they can therefore make antibodies against these antigens. So class 1 is HLA A, B, C, and class 2 is HLA DP, DQ, and DR. Out of these, A, B, and DR are non-negotiable. You have to go for HLA typing for these, especially in renal transplants and liver transplant. But in solid organ transplant, you have to go for the full house matching, that is ABC, DP, DQ, DR. So basic structure of HLA class 1 and class 2, it has been very exhaustively explained in the earlier episode on overview of HLA. But just to go through the same, class 1 and class 2, there are basic differences between the peptide binding group. You can see the class 1 is formed from alpha 1 and alpha 2, whereas class 2 is formed from alpha 1 and beta 1. Class 1 is a monomer, whereas class 2 is a heterodimer. You can see there are two chains here. The transmembrane chain also, you can see there is only one chain and there are two chains in the class 2. Beta 2 microglobulin is covalently bonded in class 1. You have this separate chain, and here you don't have a beta 2 microglobulin. Most of the antigen presenting cells, it is found in most of the antigen presenting cell, the class 2, whereas in class 1 it is in all nucleated cells of the body, and along with that the platelets also express class 1 antigens. Coming to the basic technology which we all talk about, what is Luminex? So Luminex is the buzzword right now. It is the technique on which we perform the most sensitive HLA transplant immunology workup, both for HLA typing and HLA antibody testing. So what is Luminex? So Luminex, it's a technology, it's a patented technology from Luminex, which consists of several differently colored polystyrene beads. So you have these beads, these are synthetic beads which are impregnated with dyes. So these dyes are fluorescently labeled dyes. So depending upon, suppose you add two dyes to the bead and you have different concentration of each dye, you will have a different color for each bead. Suppose you can alter the ratio of the two dyes or the fluorescently labeled dyes to have different beads altogether. So you can have hundreds of beads with different proportion of each of these two dyes. So once you have these dyes, you have a unique bead which is specific to those dye. So for the Luminex bead technology, you have several hundreds of these beads on which an HLA antigen is coated. So suppose this is one bead, this is second, this is third. So they are different colored beads, and on each bead you have an HLA antigen coated, to which a corresponding antibody will bind when you add the serum. So if a patient contains the anti-HLA antibodies, it will bind to those antigens on those specific beads and react with each other. So but how do you visualize the same? So you need another antibody to visualize, which is generally a phycoerythrin labeled anti-human IgG antibody. So this is also fluorescently labeled. So basically you have two dyes here to make a unique bead, and the third dye you add with the secondary antibody to visualize what is going on. So as the beads pass through, it's like a flow cytometer sort of thing. As the beads pass through the channel, they are hit with lasers of specific wavelength exciting both the fluorescently labeled secondary antibody as well as the fluorochrome of the bead itself. The bead itself has two fluorochromes, like I told you. So just to go deeper into the same, so you can see there are sets of beads here. They are all unique in the sense that they have different concentration of the fluorochrome. You can see that the color is changing. It is going from lighter to darker depending upon the concentration of one bead to another. So these beads are impregnated with a dye mixture. They visualize the cell of the body. I mean, you can see that they are trying to simulate a cell of the body with different concentration of bead. Now on these bead there is an HLA coated, HLA antigen coated. So when this bead with the HLA antigen coated, and along with that when you add the serum and antigen antibody reaction takes place, it runs through a channel. It is hit by two lasers. Now what is the function of these laser? It is to act on these beads and it will cause fluorescence, and that can be read as mean fluorescence intensity, or MFI. So when an MFI is generated, it's like a numerical semi-quantitative value. You can assess whether the bead is actually, with the bead which is there, is actually positive for anti-HLA antibody or negative for anti-HLA antibody. So these are basically one of the most important solid phase assay. Others are ELISA, which are not being used nowadays, but Luminex bead assay and flow cytometers are used very widely in case of transplant immunology test. So these are bead based assay. I have told you already. Polystyrene beads are the beads that are impregnated with the different ratios of two fluorescent dye, and this is known as a classifier signal. So when the laser hits on these fluorescent dyes, it gives a classifying signal, and you can distinguish almost 100 bead of 100 distinguishable bead population can be distinguished here. So this is one, and secondly you have a secondary antibody which is coated, and this is labeled with the third fluorescent dye, which is the reporter dye. This simultaneously assesses the HLA specific antibody binding. So this will tell you about the bead itself with that HLA antigen which is coated, and this will tell about whether the antibody is present in a specific strength or not. Just to simplify the same, this is the bead which is impregnated with the dye. You have an HLA molecule which is coated on the same. When you add a serum, and if the serum is against this HLA antigen, you will have an antigen antibody reaction. You add a secondary antibody which is fluorescent labeled, which can be detected through a laser. So the readout in the conventional flow cytometry is the mean channel shift, whereas in a Luminex fluorocytometers, you have two lasers like I told you, and the readout is a MFI, or mean fluorescence intensity. So what is the single antigen bead assay basically? So like you have a Luminex beads, each bead is coated with just one antigen. So when you have one antigen and you have an antibody against specific that one antigen, then it is known as a single antigen bead assay. For example, suppose you have an antigenic makeup of HLA A02, and because of the transplant or transfusion or pregnancy, which are the three ways where you can produce antibody, an anti-HLA A02 combines with you. So what you have detected is anti-HLA A02, which is specific to the A02 antigen that you have. So that means that single antigen has been detected. It's not a group of antigen. You have actually narrowed down to, okay, my antibody is against this anti-HLA A02 antibody is there. So that is how you tend to, it is more specific, and suppose that comes positive, you can change the donor if you want. So you don't want a donor specific antibody because it is present in the donor, so the donor antigen, and you have an antibody, so you don't want a graft when you have a DSA. So you can change the donor, or you can desensitize the patient. Basically, if you have a MFI values of, let's say, more than 1000 and you want to reduce it, so once the MFI levels or the antibody levels decreases, you can go ahead with the transplant. So single antigen bead assay allows identification of HLA antibodies for all common and many rare antigen and alleles up to 11 HLA loci. It is a very rapid test. The value comes in 3 to 6 hours. Very high throughput. You can have 96 tests at one go. Additional multiplexing ability to test many patients simultaneously. So this is how a graph would look like when you have a single antigen bead assay. You can see you have basic cutoff lines here in orange, red, yellow, and green. So this means that you have, this is the degree of on the in this axis you can see there is a degree of fluorescence, which is the mean fluorescence intensity. You can see that this antigen, specific antigen, has the maximum MFI, and in the decreasing order you can see that the MFIs are decreasing. So, but the important thing to remember here is you some MFI will be given by each bead because they have their inherent this dye, fluorescent dye. So they will give out some bead, but the idea is that you should have a MFI values above the cutoff that you have for your own lab. So it's a semi-quantitative thing. Your lab may have a cutoff value of say 500, the other lab may have a cutoff value of 1000 MFI, depending upon whether you have a liberal or a conservative approach. So, I mean, both are okay, but then you have to be consistent with it. So, for example, generally the cutoff that is taken for class 1 and class 2 is 1000. So, anything above 1000 MFI is considered positive, and anything below 1000 is negative. So you have to have your own standardization. Every lab does that for its own lab. You know, you compare it with the flow cytometry cross match or other techniques, and you derive a way that okay, this is the MFI above which I'm going to give positive. So the what is the advantage of Luminex? Luminex technology is shown to be very sensitive, especially when you compare it to other assays like complement dependent cytotoxicity and ELISA assay, and it allows you to accurately evaluate the sera containing complex mixture of antibodies. So you may have multiple antibodies, but you can detect it to the level that you can say that it is at this antigen, against this antigen this antibody is made. So it has beads containing different HLA-C, DQA, DPA, DP beta. All alleles are available, and you even have a single antigen bead assay of MHC class 1 chain alpha, which is MIG-K. So you can do that, and this basically reduces the number of false positive because the antibody is binding to the HLA antigens which are coated on the synthetic beads. So, but is that all? I mean, you have single antigen, so why do we still go for other tests? And is there a need for other tests, the more specific yet not so sensitive tests like CDC, antibody screening, and other tests which are which are complementary to this virtual cross match or single antigen bead assay? So what is the interpretive consideration? Is it the final answer? Well, no. I have already told you it's a semi-quantitative output. The MFI of one lab may be different from other. The MFI of one locus may be different from other. So generally what we do is in HLA class 2, you can be more liberal compared to HLA class 1. HLA class 1, we are very rigid with the MFI. We anything above 1000 we can give it positive. But HLA class 2, when you make a cutoff of let's say 1000, sometimes it may lead to false positive reaction. So you need to be a little liberal there, and so class 2 basically 1000 to 1500 can be the MFI by which can be set at your own lab. But there are other labs which will like to keep the MFI very conservative to let's say 500, and anything above 500 they would like to say it uh, positive. So it's your own lab and how you have standardized it, what kind of a background you have in your lab, whether the tests have been conducted by the experts. So that is very important, and that's why MFI values are very subjective, and they can be they can't be compared with every lab. But yes, in one region you can have your EQUAS program and you can compare the MFI values. But still, nonetheless, MFI values have to be interpreted with caution, and many laboratories validate their SAB assay or the single antigen bead assay with known negative and positive sera. So the threshold has to be made after you do a quality check, standardization, comparing. So then only you can start reporting out for your MFIs. Now, the other problem with the MFI is ideal scenario is when you have exact antigen, because it's a synthetic bead, you are coating the antigens, the HLA antigens to the same. The ideal scenario is when you have exact antigen strength, and on that the antibody will come and you will have an exact MFI. But sometimes in one from one suppose vendor to another, you may have a decreased antigen sensitivity from one locus to another, from one vendor to another, from one lot to another. You may have some decreased antigen density, and if your antigen is less, the antibody will coat less, and that's why you can have a negative MFI when it is actually positive. And sometimes you may have a non-specific background. I mean, your antigen is the same, but your antibody that is coating is not the antibody, but there's a lot of background. The non-specific antibody is coating the antigen. So you can have a positive MFI when it should not be there. There can be some interfering factors, or sometimes you may have a sharing between the two epitopes. We all know the antibody is formed against the epitope and not antigen. So you may have some public and private epitope. Private epitopes are the epitopes that are restricted to that specific antigen, but public epitopes are the epitopes that are shared between the two antigens. So I mean, the antibody may come positive for one, and it may also have a cross reaction with another antigen, which may be having the shared epitope. So these are the problems that are generally should be kept in mind when you are interpreting the MFI values. Other very important thing is the Poisson phenomena. So we know that there is a zone of antibody excess and a zone of antigen excess. So only when you have the exact equivalent zone where you have both antigen and antibody in equal ratio will we give the reaction. Many a times your test may be negative initially, but that is not fitting the things that is clinically seen. So what you need to do is you need to dilute the sample, and sometimes it may give positive, and that is because your antibody is so excess that sometimes it may, because of the Poisson phenomena, not give you the positive result. So this is another this is a paper that we had published, and this is also shows you one fallacy of a virtual cross match. The fallacy is that the virtual cross match depends upon synthetic beads. It's not the real lymphocytes of our body, and these synthetic beads are coated with uh, the antigen. So when the antibody reacts with this antigen, it comes positive. But these synthetic beads sometimes can lead to some cryptic antigens, which are not really the antigens that are present, and sometimes they may get exposed because of the reagents that we are using. So in many occasions, everything else is coming negative. The other tests are coming negative, but your single antigen bead assay is coming positive. So that is where the role of the controls are is very important. So you need to keep that in mind. It's not like your single antigen bead assay is positive, and I mean you can report that in isolation. You have to take everything into account. If your single antigen bead assay is positive, obviously your HLA antibody screen will be positive. Your CDC may, or a complement dependent cytotoxicity test may or may not be positive. So you have to keep that in mind. So there are many interpretive consideration of solid phase assay. It is a semi-quantitative output. I have discussed pros on the antigen density between the loci may be different. Suppose HLA-C and DQ, which are always underrepresented, but because they are not that polymorphic, they are polymorphic, but not as polymorphic at HLA-ABC. So you need to keep that in mind that the value of HLA-C and DQ may be lesser. The MFI values may be lesser than the other alleles. Then there may be conformational change. Like I said, there are cryptic epitopes. We had published a paper on the same. Epitope sharing we have discussed. And there are some modifications in the lab which always, techs would like to do. Sometimes you might like to, take out more tests from that same sample. You may modify the temperatures or the dilutions. So you have to keep that in mind. It is very locus specific. We have already discussed it is lesser in HLA, C and DQ and more in ABC and DR. So you have to keep which loci you are reporting, and reagent and lot specific as well. So one lot may not give you the same MFI values as the previous lot. So you need to always, put the controls when you start running a new kit. So what is a virtual cross match? We have talked about single antigen bead assay. It is very simple. So virtual cross match means you are not actually physically performing the cross match. It's not like pro cross match or CDC cross match. It's a virtual cross match. So what you are doing is it essentially means donor cells react with patient serum, but you are not actually doing it. So donor cells means donor antigens. Donor antigens mean donor HLA antigens. So donor HLA antigens means you have to do a donor HLA typing, and you see what HLA typing the donor has, and then you compare it with the patient serum which has antibody. So suppose a donor has an antigen of A02 and you have a patient who has an anti A02, then obviously your virtual cross match is positive. So it is the it refers to the comparison of the anti HLA antibodies of the recipient, which is defined by the Luminex, and you compare it with the HLA typing or the antigen of the donor. So donor cells and patient serum. So if there is a donor specific antibody present, it would be positive virtual cross match. If it is not present, it is a negative virtual cross match. So coming to questions and some real life case scenarios, just to highlight the importance of single antigen bead assay and the virtual crossmatch, and some interesting faces. So you can see in this, this is a worksheet that we received on this patient who has a donor. This is the patient single antigen bead assay, and you can see that the donor HLA typing is HLA A0202 3601 and HLA B1510 and B5801. If you can't understand this, you have not seen the first video that we had on basics of HLA. I would request you that you go back and see that HLA overview, and then you would understand the nomenclature of the HLA, and then only you can interpret the actual report. So you see the donor HLA typing is HLA A0202 3601 and HLA B1510 and 5801. So what you do is, now you have the donor typing, you do an antibody single antigen bead assay for the patient, and you see which are positive. You can't, I mean, it's not very clear here, but till this 137 bead here, it is positive. Why it is positive? Because the MFI values is more than 1000. So here you can see it is 1254. So anything above this is positive. Now whether it is a donor specific or not donor specific, you need to compare the HLA typing of the donor. So you can see that it is positive for B15. It is B1518, B1512, B1501, B5701, B1513, B1302 and B4501. So you can see none of this is here, but it does have, the donor does have HLA B1510, but the B1510 is not even there in the whole antigen profile of this single antigen bead assay. So the point I am trying to make here is, your single antigen bead assay will have around, let's say 100 beads, but you cannot cover all the HLA antigens that are there which have been discovered, because there are thousands of HLA antigen. It is hyper polymorphic, you can't cover everything. But what you can do is, loci wise, if you see, just disregard the suballele. B15 is positive. All the 3-4 beads of B15 are positive. So most probably B1510 will also be positive. But since it is not there in this kit, you cannot comment on the same. But that doesn't mean that you do not give the report as positive, because once this patient goes for a transplant, more often than not, he or she will have a rejection. So in contradiction to the sensitivity of SAB assay, patient may also have a DSA for a bead not present in the bead blend of the test kit. So this B1510 is not present here. So you can't comment whether it's positive or negative. But what the clue there is that all the B15, whether it's 16, 12, 01, 02 are positive. So most probably B1510 will also be positive. So this is how we report, although bead for 1510 is not present, but can be considered positive for B15 with an MFI value of 3473, which is above 1000. So we give it positive. Similarly, second case, donor HLA typing is HLA-A 1101. This is homozygous for 1101. HLA-B is 1502. So there is an allelic difference between the HLA-B. So these are two different allele. 1502 is very different from 1503, and we have an HLA-C 7040801. So after comparing, we saw that it is none of these antigens are present here in the positive range, but what is present is C is positive and C07 is positive at various ends. So C701, then C0702, C0801. So you have one donor specific antibody C0801, that is for sure. C0801 is here in the donor, and you have an antibody of C0801. But what is also is important is C0704 is not present here. So there is no representation of C0704. But C0701 and 02 and 03 are positive. So the report that I would like to give is donor specific antibody detected against B1503. You can see that B1503 is here, and it is also positive at an MFI of 718. The second thing I would like to say is C0704 is not present, but can be considered positive for C07 because all alleles are not represented. Here only C704 is there, which is not there in the bead assay. The third thing I want to highlight is cryptic epitopes. It's a case report that we had presented. We have a paper on the same. In this, there are occasions when the tests performed before considering the patient for the transplant are ambiguous and they require a further workup. One such condition is the presence of virtual crossmatch. We had an 82601 in this case with a negative CDC, Luminex, antibody screening, flow crossmatch. Everything came negative, but only the single antigen bead assay was coming positive for virtual crossmatch. And then we also saw that all the other beads were also coming positive. Many beads were coming positive, which were actually negative when you saw the flow crossmatch and other crossmatches. So we tend to look into the case. So you have to report everything in the gamut of the armamentarium that you have for testing. So it's not like you report just on single antigen bead assay. You have to see CDC crossmatch also. You have to see Luminex crossmatch also. You have to see flow crossmatch also. If you are performing all these things, antibody screening is a very vital tool. So if you have an antibody screen which is coming negative, but your single antigen is coming positive, there is some discrepancy. You need to check that. So the way forward is the flow crossmatch is there, Luminex assay is there. These are a very sensitive test, but at the same time we have not so sensitive specific tests like CDC, which is still being performed. It's a necessary evil. Many call it the in vitro transplant. Then there are other cross matching techniques, but single antigen is also there. Virtual cross match is there, but this is not the final or the only answer. You have to have a battery of tests that you employ. Suppose say like two cross match, one can be a virtual cross match, one can be a cell based cross match like a CDC cross match with a flow cross match. Antibody screen is very important to perform because antibody will tell you whether there is presence or not presence of antibody, whether it's donor specific or non-donor specific. That is a different case. So single antigen bead assay is most sensitive test. You can actually know the specific antibody and then you can act on the antibody and cause desensitization. So single antigen bead assay should be performed wherever necessary, but it requires careful interpretation, and you should understand that it is not the final answer and you need to know the limitations of single antigen bead assay. We should have a test algorithm for compatibility to increase the sensitivity and the specificity of the testing process. So you can have a battery of tests or that this is what I'm going to employ initially, and then if this comes positive, I will go for this test. So for our own country, we have a multicentric study which is called the Delhi approach, and this shows you the algorithm for HLA antibody testing in renal transplant patients, especially solid organ transplant like renal transplant. And you can go back and refer to this Delhi approach if you want to simulate the same. Otherwise, you can develop a own report or own algorithm based on the test that you have within your system. So if you have a flow cross match or say Luminex based, lysate based cross match, so you can perform either or. So of course, if you have all the things or all the tests, which is very unlikely, you can perform all. And plus, if your patient is affording enough to do all the tests. For a cost constrained country like ours, we need to have an algorithm, and single antigen bead assay is very vital for the same. So I hope you have understood something about the virtual cross match, single antigen bead assay, and the need for an algorithm testing for compatibility, especially the histocompatibility testing. If there is anything that you have not understood or want me to explain again, you can always write in the chat box. Thank you so much. I hope this helps.